Good morning, everybody, and welcome to this lecture on the angiotensin-converting enzyme inhibitors, also known as the ACE inhibitors. Now, these drugs are specifically used against hypertension, and they inhibit this ACE enzyme. Now, the mechanism of action in which these ACE inhibitors then block the angiotensin-converting enzyme is given to you here at the bottom of my slide where I've narrated it. Please also make sure to watch the introductory video, which explains the process of how hypertension happens from these ACE enzymes, as well as the drugs that can inhibit the ACE enzyme and help against hypertension. Then the drugs that are currently used as ACE inhibitors are also known as ACE inhibitors. And they reduce the production of angiotensin 2 to angiotensin 3. So they basically inhibit that process. So they inhibit the ACE enzyme. The drugs include captopril, lisinopril, enalapril, and so forth. Now, if you, for instance, now look at the chemical structures of all of these compounds, you will already note that there are tertiary amines. I see a tertiary amine there. I see a tertiary amine here. I see a tertiary amine here. We also see that there are carboxylic acids in all of these structures. Interesting to observe as well is that some of these carboxylic acids are not actually carboxylic acid, but they are esters. So that could already give us an indication that these or some of these compounds are actually prodrugs, because we know that esters can be converted enzymatically in the body by esterase enzymes into carboxylic acids. So already by looking at the different chemical structures of the different ACE inhibitors, we can already now see certain structural features that could give rise to structure-activity relationships. And this is what I asked you to do here at the bottom of the slide in my notes. Then this is just some more background on how the ACE inhibitors work. Again, make sure to go through your introductory video. This is some more background information about the physiology of the renin-angiotensin-aldosterone system. I've even put it in this slide here to try and explain to you this schematic. Please make sure to go through this. This is for background so that you understand the physiology as well as the pharmacology around the ACE inhibitors as well as the renin-angiotensin system. There may be one or two MCQ questions in the tests or on the exams on the mechanism of action of the renin-angiotensin system. Now, back to the actual ACE inhibitors. The ACE inhibitors are enzyme inhibitors, the angiotensin-converting enzyme. So it's an enzyme. So with the enzyme inhibitors, you get IC50 values. So you have an inhibitory concentration where the drug then blocks this ACE enzyme by 50%. And the way that the ACE inhibitors work is that they go and bind to an area within the ACE enzyme, the active site. And we can already see here, this is the active site of the ACE enzyme. And then deep within this ACE enzyme, we see that lisinopril, which is an ACE inhibitor, can bind to the active site, or also known as the substrate binding site, because that's the same site where the ACE substrate binds. And if we look at the closer view, you can clearly see that lisinopril fits nicely into the active site, and it does potentially have interactions with amino acids surrounding the active site of the ACE enzyme. And if we look closer, this is one example of captopril that has been docked into the active site of the angiotensin-converting enzyme. And you can clearly see here that there's certain binding interactions. Like, for instance, in this case, on the carboxylic acid, there are two binding interactions. The one binding interaction is, it looks like a hydrogen bond interaction with tyrosine 520, with tyrosine 520, as well as one ionic, pi-ionic interaction with histamine 353, as well as a zinc binding interaction with the sulfhydryl group binds with the zinc binding site within the angiotensin converting enzyme model. So just by looking at the docking and the binding interactions, it could give us more information on what is necessary for these molecules and part of the pharmacophore. If I just look at it, it seems as though the carboxylic acid is important, that sulfhydryl group is important, and possibly this group over here, this amide group over here as well, could be quite possibly part of the pharmacophore. Now, if we then actually go to the pharmacophore of the ACE inhibitors, you will note that the pharmacophore is basically where you have a carbonyl group at this position, an amide, as well as a carboxylic acid. So here we have minimum structural requirements. And indeed, we need a carboxylic acid for ionic binding interactions with the enzyme. We need the amide oxygen that forms hydrogen bonds with the ACE enzyme, as well as a functional group that can be any functional group that is a sulfhydryl, phosphor, or a carboxylic acid group on the R1 position. So it's this position over here of the pharmacophore that binds to the zinc binding area of ACE. I've shown you the zinc binding area for captopril. Captopril, that sulfhydryl group, forms an interaction with the zinc binding area of the angiotensin converting enzyme. So please take note that the ACE enzyme has an amino acid in its active site, and in close proximity to that amino acid is actually zinc, a zinc molecule in the enzyme. And the ACE inhibitors can then form an interaction with the zinc binding site, and thus give it then the inhibitory activity. I've tried to show you here the different functional groups, the sulfhydryl functional group, the carboxylic acid group, as well as the phosphor functional group that can make part of R1 position of the pharmacophore of the ACE inhibitor. So if we look at captopril, let's see if it fits the pharmacophore model. Yes, we see a carboxylic acid, we see the tertiary amine or the amide, the carbonyl oxygen, as well as a functional group, in this case sulfhydryl, that can form a binding interaction with the zinc binding site. Even with lisinopril as well, lisinopril, we see the carboxylic acid for ionic binding, we see the amide group over here, as well as we see a carboxylic acid group, in this case, carboxylic acid functional group, that could form binding interactions with the zinc binding area. So you can see that both captopril and lisinopril, as well as all of the other ACE inhibitors, abide to this pharmacophore model of the ACE inhibitors. Now, the ACE inhibitors can be categorized in 3 subgroups, namely the sulfhydryl ACE inhibitors like captopril, the phosphor ACE inhibitors like for lisinopril. You can see for lisinopril as this functional group over here, that is the phosphor functional group. And then you have a few drugs that are dicarboxylate ACE inhibitors, where at the R1 position, it's a carboxylic acid, but they have another carboxylic acid, like for instance, if we look at, let's look at quinapril. So quinapril has a carboxylic acid in this position over here that can form ionic interactions as well. It has this carboxylic acid group on this position here that can form interactions with zinc. So quinapril is a dicarboxylate ACE inhibitor. But look carefully at the structure of quinapril. Is that a carboxylic acid? So think about that. Is that a carboxylic acid? We look at the molecular formula of it. It is COOC2H. It looks like a 5. H5, yes. So do you think that that is a carboxylic acid, or is that an ester? So that is an actual fact, an ester. And what have we learned from esters in our prodrugs? Esters can be converted by esterase enzymes into an active form. So yes, quinapril is a prodrug of the ACE drugs. So quinapril on its own is not active yet. It first has to be activated in vivo into its active form. So by esterase enzymes, this this ester will then be converted to the carboxylic acid. So please take note of this. Quinapril is inactive. It first has to go in vivo esterase activation of that ester group into the carboxylic acid before it will then form a binding interaction with the zinc binding site. This is very important to note. If you, for instance, give this to me in the exam and tell me that quinapril can perform a binding interaction with the zinc, you will lose your marks. You first have to show me that quinapril will be activated by the esterase enzymes into a free carboxylic acid group that can then form the binding interactions with zinc. Similar for ramipril. Ramipril has the carboxylic acid, but it's an ester that first needs to be activated. It's a prodrug, activated, then it can interact with the zinc binding area. The same thing goes for benzopril and enalapril. All of them are prodrugs of the ACE inhibitors. So please take note of that as well, especially for the dicarboxylate ACE inhibitors. But let's, for instance, look at lisinopril. Lisinopril has the 2 carboxylic acids, but you see that it's not an ester. So lisinopril is a drug that is already active. It can form the binding interaction with the zinc binding area because it doesn't have to be activated into its active form. It is not a prodrug. Captopril is also not a prodrug. So it's only enalapril, benzopril, quinapril, ramipril, and lisinopril that contains the ester that gets converted to the carboxylic acid. There's just one correction that you have to make throughout my slides. Please take note that ramipril, it's missing a tertiary amine. There's a tertiary amine that needs to be in that position over there. So please make a note of this and update all of the ramipril structures within my notes as well as within my slides to have that tertiary amine on the position that I've just indicated here. Then the ACE inhibitors are also are not very stable. They have stability issues. They may undergo hydrolysis when it gets into contact with water. And a lot of times this hydrolysis becomes more rapid when it's also exposed to acids, bases, heavy metals, and high temperatures. So that's why whenever you see a ACE inhibitor, if you dispense it in a pharmacy, if you look at its packaging, if you look at its labels, its PIL and PI, you will see that there's always a statement, store away in a cool, dry place away from direct sunlight. And the reason for that is because if the ACE inhibitors get into contact with water, and especially at higher temperatures, then it undergoes degradation and the molecule is no longer stable. So let's look at the stability. So the main thing that causes these stability issues is this amide group within the ACE inhibitors. Like we've learned in the prodrugs as well, esters can undergo hydrolysis much quicker, but amides can also undergo hydrolysis, but it's a slower process. But in this case, with the ACE inhibitors, when there's water as well as a catalyst, then this process can actually be quite rapid. So when the ACE inhibitors are getting into contact with water, then water or moisture for that matter, then the drug undergoes degradation and 2 degradation products form. So for instance, if we look at this amide, so that amide bond gets broken at that bond. And what you then have is basically the carboxylic acid and a secondary amine. So this is basically what we've seen in prodrugs as well. Whenever you have a carboxylic acid, that carboxylic acid can react with an amine. So the same thing has happened here. It's just the reverse, where water has broken this bond to give you the carboxylic acid as well as the free amine. So these molecules, obviously, these degradation products, they do not fit the pharmacophore model of the ACE inhibitors anymore. So if the drug undergoes degradation and is not stable, gets into contact with water, you basically have an inactive pharmaceutical agent. So that's the reason why the ACE inhibitors are protected from light and moisture in its packaging material. Then, for instance, if we just go and have a look at lisinopril, what would be the degradation products of lisinopril? Well, we see that tertiary amine there. So that bond over there will be broken. On this side, you will have a functional moiety where that then becomes an NH group because the bond is broken. And on this side of the molecule, you will basically break that bond, converting it into the carboxylic acid. So those are the 2 degradation products that forms for lisinopril. So I did note it down here. Hydrolysis of the ACE inhibitors at the amide functional group gives rise to a carboxylic acid and an amine. So this is for all of the ACE inhibitors. Even for ramipril, please note that there's a missing tertiary amine here, can break at that bond, as I'm indicating here can break on that bond, forming thus the amine as well as the carboxylic acid degradation products. This is actually quite important for the exams as well. You will have to know and go and memorize this general reaction scheme for the stability and the degradation of the ACE inhibitors. Please take a note of that. I try to also explain it to you here at the bottom of the slide. Then further stability of the ACE inhibitors, and we've already discussed it, that some of the ACE inhibitors contains an ester. Like for instance, enalapril contains an ester at this position. And we know that enalapril is an inactive drug. It first has to be activated in vivo by esterase enzymes to form the dicarboxylate of enalapril, so that we now have a free carboxylic acid group that can form the interaction with the zinc binding site. So this is something that we've discussed. Remember, this is the active form of enalapril. This one here is an inactive form because the ester cannot form the binding interaction with the zinc binding site. Only the carboxylic acid, the converted free carboxylic acid, that can interact with the zinc binding site. So I have a further question for you. For all of the drugs or the ACE inhibitors that contain the ester, you have to now show and illustrate the activation, like I've done for enalapril, for each of these drugs, and then also then just show that the carboxylic acid is what is converted for the drug to be active. This is also quite important for the exams. No, not necessary to go and memorize any of these structures. I will lead you into the question. I will give you, for instance, the structure of enalapril, and I will ask you what are the issues with stability of enalapril, and then you can maybe show me the actual stability for the degradation, as well as there are some stability for the carboxylic acids, but you can say that this isn't a stability issue, this is more of a prodrug thing, where the drug needs to be converted into the active form. Then another very important aspect of the ACE inhibitors, they are chiral and they are stereoselective. So all ACE inhibitors are chiral and stereoselective. Like for instance, if we look at the case of captopril, captopril has two chiral carbons. It's the SS captopril as well as the RS captopril. But the RS captopril shows no ACE inhibition, whereas the SS captopril is active. And this is actually true for all of the ACE inhibitors, where each and every ACE inhibitor needs to be in the SS stereochemical form. And the SS form is at the 2 position, where we have the methyl group, and then at the cyclic structure that has the tertiary amine at that carboxylic acid also needs to be in the S stereochemical form. So this may be a good opportunity for you to go and revise some of your stereochemistry notes. An important tip for the exam, just make sure that you are able to assign absolute configuration. So maybe go back to the stereochemistry notes and make sure that you can number the groups according to their priority, and then also then look at the rotation of your absolute configuration. So is it, if the rotation is to the left, then it is S. If it's to the right, it is R. So please just make sure that you can still assign the absolute configuration to the different compounds, especially for the ACE inhibitors. Then I've given you some background again on the ACE inhibitors, maybe just for practice. See if you can identify the R and the S form of this drug, and even with chloramphenicol, sorry, that's not spelled correctly, so please just make that correction. You see with chloramphenicol, you have 2 chiral carbons. Please assign these chiral carbons either R or S, just as practice, so that you are adequately prepared for both the test as well as the exams. Here is some more examples of naproxen, of ibuprofen, and I've even shown you the case again of the thalidomide disaster and the importance of stereochemistry if we look at thalidomide in this case. Then furthermore, we have structure-activity relationships. We know that the ACE inhibitors are stereoselective drugs. The reason why it's stereoselective, if you look at the active site mainly consists of L amino acids. And for these ACE inhibitors to bind to the L amino acids and give rise to its inhibitory properties, the drugs need to be in its SS configuration. Or, for instance, as seen in captopril, captopril only has one chiral center. So that chiral center needs to be in the S configuration. The dicarboxylate inhibitors has two chiral carbons, and those are then in the SS configuration. And those configurations is necessary for the drug then to bind to the L amino acids present in the active site. Then there are some further structure-activity relationships. I think we've already discussed most of them. Number one, you need a carboxylic acid for ionic bonds. You need the amide carbonyl group for hydrogen bonds, the sulfhydryl, phosphor, or carboxylic acid groups to interact with the zinc binding area. You need the proline group. So proline group is this group over here. So that proline group is specifically included so that the drugs are stereoselective, so that you can get the carboxylic acid in its S form. So you will see for the majority of, or actually for all of the ACE inhibitors, you see this type of proline cyclic structures. Some of them are a little bit different, where you have the proline, but then it's connected to another aromatic, or in some cases, the proline has an additional carbon, making it a 6 atom cyclic ring. So, but in general, we need this type of cyclic structure to have the carboxylic acid and then be able to produce it in the S form. Then also, for instance, for captopril, you will note for captopril, we have the sulfhydryl containing functional group. And this sulfhydryl group is known to produce a high incidence of skin rash and taste disturbances, as well as the sulfhydryl group can cause a shorter duration of action of the drug, so shorten the half-life. So if you want a quick acting and short acting ACE inhibitor, then you can use captopril. But if you do see that the patient is suffering, for instance, from eczema or other skin rashes, psoriasis, or whatever, then obviously it would not be good to prescribe this drug to the patient, because it may cause more severe skin rash reactions. And even with the taste disturbances as well, for some patients, it can be quite a problem if there are taste disturbances as well. But these are just for the sulfhydryl containing compounds. The phosphor and the dicarboxylate drugs or ACE inhibitors do not have these problems. So it's only for the sulfhydryl containing drugs that have a high incidence of skin rash, taste disturbances, and it may shorten the duration of action. Further to the structure-activity relationships, this is just a schematic that I've drawn, a simplified schematic of how the ACE inhibitors bind to the active site of ACE. And this is just, again, captopril, and you can see the sulfhydryl group with the zinc binding area. The amide group with the carbonyl here has a hydrogen bond interaction. That's true for all of the ACE inhibitors. The carboxylic acid can form ionic interactions. And then, and remember, the carboxylic acid needs to be in the S configuration. And here you have the methyl group that's also in the S configuration. It's close proximity to lipophilic amino acids, and it can then form the lipophilic binding interactions. The proline group is also known to cause lipophilic interactions. If we look at benzopril, for instance, that proline group, it's not actually a proline group, but it is a group that contains, it looks like 7 atoms. But that group still can form the lipophilic interactions. And you can even see with, for instance, enalapril, you have the carboxylic acid to form the ionic interactions, and you have the methyl group on this position to form the lipophilic interactions that needs to be in the S configuration. What I would like you to do as an exercise for both the test and the exam is to use quinapril, ramipril, enalapril, and benzopril, and to redraw each of them as I've done for captopril. That will be a very nice exercise to adequately prepare you for the test and the exams. I did make notes of this, of what you need to learn for this specific section. Then we also have some specific ACE inhibitors and some more information about the ACE inhibitors. This is just for noting. Please go through this in your own time. And here, basically, I'm also talking about specific ACE inhibitors and those ACE inhibitors who are actually prodrugs, like, for instance, enalapril, ramipril, and fosinapril. And we've discussed this at length. So that brings me to the end of my presentation. I hope you all have enjoyed it, and I've given you hopefully some more information to think about and be better prepared for the exams and tests. I wish you good luck. Thank you.